Within multicellular organisms, tissue displays high degrees of order in their spatial organization. Cells align and display oriented polarity over large distances over the course of morphogenesis. To understand how the planar arrangements of epithelia is reached during development, it is crucial to track cells' orientations and growth dynamics with high spatiotemporal fidelity in vivo. The described protocol is designed to image and analyze cell behaviors at global and local levels in the Drosophila pupal epidermis.
This methodology could provide insights into different areas of research, cell biology, morphogenesis, and planar polarity, and can be applied to the analysis of other tissues, structures, and models. All the steps can be performed easily after some practice. They demand precision. Visualization of this protocol helps achieving optimal results in a shorter time. The pupa dissection is complex.
To begin, use a moistened paintbrush to transfer staged white prepupae to a fresh plastic vial, and keep them there for as long as necessary at 25 degrees Celsius. Then, remove each pupa from the wall of the vial with the help of forceps. Glue the ventral side of each pupa on a glass slide covered with double-sided sticky tape. Gently tap on the head spiracles and the dorsal surface of the pupa with the tips of the forceps to assure the adhesion of the pupal case to the tape. Begin dissection under a stereo microscope by gently removing the operculum from the puparium with the forceps. Insert one tip of the forceps in a shallow angle between